So as Dr. Pritchard mentioned in his talk, NGS reporting is still a very labor-intensive process. We need to weed out these false positives, and that even these latest, fanciest sequencers and bioinformatics pipelines are producing. So, but if a human can tell that the difference between a technical artifact and a real disease mutation, I think with enough effort we can start teaching computers to do the same, either by coding those direct solutions or through artificial intelligence to help them learn themselves. So this is not going to replace the pathologist, but it's going to make their job a lot less frustrating. And so Dr. Jochen Lennerz, the keynote speaker for the computational pathology half of the symposium, has been at the forefront of this research and development, and why we invited him to speak today. He's double boarded in pathology and medical genomics, genetics, and is currently the associate director for the Center for Integrated Diagnostics at Mass General, and associate professor at Harvard Medical School. So I'm really excited to hear about his research today. Please submit your questions in the Q&A portion.
All right. Hope you guys can hear me. Can you see my screen?
Yes.
Or is this the shared screen again, the trick here? No? All right. Thank you very much for the kind words, Nathan. And this is a perfect setup by Colin and Andrew. Thanks a lot. I have no conflicts of interest, and I wanted to briefly outline what is actually the key question here. Obviously, you know, the talk title suggests that I'm talking about AI in clinical practice. But I think the bigger theme here is how to integrate such novel and complicated diagnostics, for example, in computational pathology, into the clinical practice, and then actually how to navigate this and what are some of the paradigms that we can learn. So as Nathan said, I, with the help of a lot of people, direct the thing called Center for Integrated Diagnostics. And there's a lot of words here. I think that's much too long. But I think the key part here is integration. And I wanted to start with that before we jump into the machine learning and, you know, coding aspects and how to use AI, by briefly showing maybe some of the problems that you saw or heard in some of the prior elements in the talk. So one is you want to launch a new test, but there's a lot of time requirements. People want the results much faster and ideally clinical grade, but that is a high complexity environment. So that is basically not really compatible, but you have to make it work. Everyone wants the completely most novel and, you know, highest cutting edge or most cutting edge elements, but they're billing and reimbursement questions. You have an internal vision of what you want, but then, you know, stakeholders may not agree on the financial constraints, for example. And you have, you know, obviously a tremendous amount of data analytics to be done, but that requires a lot of resources. So how to align this? And this has, you know, been a puzzle for a long, long time, and how to keep these all in balance. I don't have a perfect solution. So there's no like one size fits all solution, but I want to show you how we tackle this. So the trick, so to speak, is to kind of think about those in three buckets, and I think you all do. This is just our way of portraying it, and I wanted to briefly show you. So we strictly separate what we do in the Center for Integrated Diagnostics from the research, the ideation or the analyte stage. Typically, we start with something that's published, or at least in clinical trial evaluation, so where we can actually really test it in clinical practice. So that is usually where it starts. And then the trick is, can we really bring this on for a sustained time, talk to payers, and really get it reimbursed to keep this change sustainable? Because if the change is not sustainable, it's not really a change. It was just sort of an adoption, but that didn't last long. So I think that is an important starting point. To briefly outline to you how we organize this is, and I want to already thank my team for this. So for example, the research lab, here's the one from John I. Frady portrayed, works in close collaboration with our administrative research core as well as clinical laboratory staff. We have a set of attendings that have a dual role. One is to review, obviously, cases and sign out as a clinical position, but also every single attending has the job to bring on new tests and evaluate new technologies. And that team is supplemented by a team of computational pathology people that are designated to help with this process and also implement some of our pipelines. So just to give you an overview and that I have data to support this approach before I get to what we want to talk about today is, if you look, for example, our numbers, so we have a total of, I mean, this is total of cases, I think since 2013, you see we do over 90% clinical work, a little bit of research, which is, I would say, close to, I don't know, clinical research as it gets. The rest is just validation, proficiency, and clinical trial. And if you look at our samples, I think this is probably reflective of most molecular diagnostic laboratories in the country, roughly an equal split between heme, lung, and gastrointestinal cancers. And then, you know, there's obviously many, many more. We don't do a lot of breast cancer testing. That is maybe one special element, but I think breast cancer is actually here, this yellow component. But to give you two additional things that I find very important when we discuss the variant reporting soon, is to show the landscape over the last couple of years. So this is an overview from 2013, when we started implementing next-gen sequencing in clinical practice, all the way to 2020, which is our current state. So this is data year to date. We have this running as a business report, just sort of for leadership to make sure that we know what's going on. And one thing that I outlined here with these lines is, as NGS went up, for example, FISH, which we use mostly for copy number and break apart assessments that we can also detect by next-gen sequencing, is consecutively going down. And you can see this even reflected here in our year-to-date. The other thing is obviously the overall volume of testing goes up. And if we portray this slightly differently in sort of a grouped stacked bar graph, NGS goes down, FISH, NGS goes up, FISH goes down. And then we put in a couple of other elements here just sort of as a comparison. I think it's very important to realize that, yes, comprehensive testing is going up in a almost constant growth rate, but the element that I find very important here, what are the implications? And as Andrew has pointed out, that creates obviously the need for more specific domain knowledge, more manual review, but also the risk of potentially missing certain things. So before we get started, I wanted to share this very, very bright red slide just for relaxation, just so you can get ready. So I'm sure if we start all the way in the background, so we have to ask questions slightly different. If you look in the background, the hands with the red and the blue pill, I don't know if you remember, but that was a scene from Matrix, where you say basically, do you want the red or the blue pill? I never figured out which pill is what, but I don't know, there's probably YouTube videos on that. What I wanted to illustrate here, overlaid by, in this case, a random forest tree, that's a decision tree, that whenever we make a decision, we don't run code like shown in the third layer on top of this here, but we ultimately say something like, do you want red or blue? And you can make those binary decisions and apply those to variant reporting. So when looking at IGV pileups, you ultimately decide one very simple thing. Will I put it in the report or will I not put it in the report? And what's illustrated here in this random forest tree visualization that I get to later on, there's probably many, many different factors that you put for or against a certain variant. For example, in this particular transcript, is the consequence, is that present or not, or whatever. So what I try to illustrate here is that when applying sort of artificial intelligence or machine learning or whatever you want to call it, algorithmic decision-making or decision support tools to a clinical decision, you work in this multi-layered land of coding that probably most clinicians want to understand, decision trees that very often I would argue we don't even understand, and in typically decisions that may not seem as simple as a yes or no, but that's kind of the background. So I wanted to show what I think Andrew has beautifully outlined in some of his slides is, once you have NGS data and they're pre-processed aligned, and at some point you get a filtered variant call file out of it, you have to perform manual review. I know that there are a lot of exome and whole genome studies out there where that is not done, and I would argue that that is of lower quality. And just taking a pipeline output, it doesn't matter if you take 10 cases or 10,000 cases. You may see trends, no doubt about that, but the quality that we apply in clinical practice when we manually review pileups is just currently not really replaceable. I mean, if you ever seen a pileup near a poly-G tract, you know that there's just scattering, and pipelines call that. You can argue, well, why? And you can spend probably half a year optimizing your code to try to get rid of it. But the much faster thing is just to do the manual verification step. And I think that is a very complicated process. So what I put here as sort of an arrow as a reporting decision is a multifactorial thing that ultimately says, yes, I put it in a report. And then once it's in the report, there's multiple steps to actually result in a clinical decision. So I want to make sure that you follow that this is only the decision of going into the report. So we talk about variant reporting, not a clinical decision support tool. So the FDA would probably call this tool that I'm about to show you a decision support tool because it is obviously a decision, but it's distinct from a clinical decision support tool. For example, find an ALK inhibitor if you have an ALK rearrangement, etc. So this big pie chart again shows the variants that we have, and you can see, I think, Andrew, if I recall correctly, you mentioned that 37% or so of variants you filtered out. So you can see here, our pipeline has different, we have also a composite caller. We can go into the details, but if you see that our pipeline spits out in this particular data set 19,954 variants from various cases. And when we look at what we don't report, meaning what humans filter out, it's 76%, and 24 actually goes into the report. And I think, obviously, this is not reflecting every single case, but over many cases and many variants, that's the case. So to show or illustrate how difficult some of these decisions are, I took two different things that I would argue are important elements that go into this reporting decision. So for example, most people who review a pileup would want to know what tumor type they're looking at. Just, melanoma has BRAF and, some other tumors don't. The other thing is you want to know what gene, by means of gene symbol, you're looking at. So if I look at a KIT gene versus a CDKN2A, I would want to know which one that is. That may not be intuitively clear in IGV unless you look at what the gene is, but the pileup or the individual reads, once they're aligned, can give you that. So let's just look at these two elements. So of this multifactorial pipeline output, we're just looking at two. And we try to symbolize what the raw pipeline output is, meaning these are all cases all variants that need to be vetted, versus those that a pathologist or a reviewing molecular pathologist says, we are not going to put this in the report to leave over those that go into the report. So this is basically raw output minus human elimination equals yes goes into the report, shown over 2 dimensions, tumor type versus genes. So as everyone on the call knows, TP53 is probably the highest prevalence of mutations. And you see this is present in the raw pipeline output. So if you would say, obviously this is a smaller panel, but let's assume this would be an exome file, clearly the trend TP53 is commonly mutated is clearly visible. And then when you look at what is actually filtered out by a human after human review of the variants, you see TP53 is still on the top of the list. Nonetheless, you see these almost like barcode looking stripes here. These are, for example, RET, FOXL2, IDH1. And interestingly, you see in this density gradient here, a lot of these are filtered out to be not reported. And thereby getting to this pattern that in some ways resembles this original one, but minus these stripes. And as you can tell, if IDH1 would be mutated across all these cancers, I'm sure it would have been reported. That's not the case. So these are artifacts that are inherent to the pipeline and where humans filter these out and remove this. So with that, I want to use this sort of as a starting point to say, most pipelines have many, many more output features than site and gene name. That would be a little limiting. But all of the output of the pipeline is discrete. So you can basically consider this, even with the binary reporting decision, almost like a neuron. Tons of input, and then the question is there an action potential coming out, yes or no? we thought that that is an ideal setting to assess the performance of an artificial intelligence approach. So if we simplify this, in the top part is the current practice. There's a human with a brain looking at a variant and decides, do I want to put this in the report? Yes, or do I leave it here? And we just took this entire, let's say, current practice variant call file, the raw file, and used this existing human decision to power a machine learning algorithm to basically just say, give me zeros and ones to kind of provide this to, or at the time of the reporting decision, as an additional input feature. So we're not replacing the decision, we're just providing this model score. Obviously, at some point, we can discuss later on whether that's possible or whether we should do this, that this is automated. But for now, we just said we want to give the human also a model score at the time of the decision. The decision is not made, the decision is supported, therefore support tool. So the first part that I needed to learn the hard way is how to actually ask these questions. So this is from Jason Brown Lee, a abstract way of formulating the problem definition from, let's say, a practical use case to, let's say, a computer scientist. So here the statement is a computer program is set to learn from experience E, which in this case would be inherent in our variant store and our prior reporting decision with respect to some class of tasks T, which is was it reported or not, and performance measure P, which is, what's the accuracy. If it performs at tasks in T as measured by P improves with the experience E. This means more here improves the accuracy as measured in P. Now this is very, very abstract. I know that there are some computer scientists here. You think, oh, that's easy. I have a very hard time going through this and really dissecting out which is a task, what's the experience, and what is the performance that I want to measure. And I want to, give you two examples that are more practical that I also had to learn the hard way. Number one is like while asking that question and building this, generally speaking, pathologists like myself say, oh, can you just build it? And then we look. And then we go back and tweak it again, but that usually takes months and months of work. And every single tiny tweak is a lot of work in terms of coding. So typically then the computer scientists ask like, why do we want to solve this? And the more sophisticated question is what needs to be fulfilled when the problem is solved. So one thing that I learned is that there's a benefit versus a use case. And I portray here two clocks to just illustrate. A benefit of knowing the time would be, am I on time when giving a talk, or will I be on time when I come to work? A use case would be to use the time to ignite an alarm so that I wake up in the morning. Those are two completely different functions. One is just sort of information input. The other one is triggering something. And I want to keep that separate. The other part is that while we have in our variant store a lot of information is, let's assume you get all the information. For example, tumor type and the gene name. And let's just say something like tumor percentage, right? Because we scrape tumors, some are 20% tumor cells, some are 80% tumor cells. Let's just say those three things. At some point, it's clear, wait, we should probably give those three things to the computer as well. And some people refer to this as trapped domain knowledge, because of course, the tumor type is not an output of the pipeline. The tumor type comes in basically with the requisition of the clinician. So in any case, so after we define the problem, we have to go through these steps. And for those who are not computer scientists on the call, I just want to briefly mention it's almost like filling out an Excel spreadsheet where you have to know the formula, you prep your data, you evaluate whether your formula worked, then you improve your results. And then once you've figured that out, you assess performance. After that comes the part that you have to implement it, which generally is not the case with Excel. But the results that you may publish or so, you would still have to implement. And then you have to build it out as a decision support tool and then actually assess whether that was really value added. So I want to show you what that looks like. Obviously, this looks a little bit different when implemented, in this case in Python. But you load the libraries, then you prep the data, which is basically meaning you sort stuff. And I get to this in a second. This is actually the core of the work. And that's frustrating, and it takes much more time than you think. At some point then, you look at the data and you parse out what is this. For example, how many primary sites do we have? How many primary site diagnoses do we have? And all of these elements here, and this list goes on, is basically the underlying, let's say, abstract representation of the data that you sorted. This is somewhat older, but I thought it was a great article in Forbes that illustrated, if you look at green, and I think, speaking about Forbes and green, obviously this is in referral to the money spent. If you look at this was from a survey that they did in 2016. If you cleaning and organizing data plus collecting data sets is 79, which is roughly 80%. So building these machine learning tools is not the fancy code and the super important, extremely high qualified computer scientists that developed 16 amazing things for very famous companies. The actual quality of the input determines the output. That is what drives the experience. And when you look at the cleaning and organizing the data, that's over half the work, I think that's a very, very important message. Now let's assume we have done that. How does this actually look like under the hood? And it took me a while to kind of get there, and I'm just showing some very, very simple overviews. So you have to encode the data. And I show one element here that is just showing how many were not and how many were reported. And I showed these numbers before to actually just sort of give a brief output. So at some point, and it doesn't matter what this looks like, but we had to encode that if someone vetted it as, we're not reporting it as 0 and we're reporting it as 1. But keep in mind, for computer scientists, even that doesn't matter. You could switch this around and grade it from 1 to 0. But we decided 0 would mean no, don't put in the report, and 1 means put it in the report. Then we, do something called a grid search, which means using various different choices of machine learning models. For example, a Gaussian distribution model, a logistic regression model, a random forest model. All of these are different mathematical functions that try to parse the data in a way that they align with this particular binary output in the best and most meaningful way. We say that we want at least 10-fold cross-validation. I won't have time going into what that means. It basically says, and now run and find in this search across all these different tools or machine learning algorithms, find the best match. Now you see after a while it says the current iteration of this is 15 of 35, and the F1 score is 92%. After the next iteration, the F1 score is 93%, and we set this, by the way, to what? And then there's another iteration, another iteration, another iteration, and then that was it. We said if after 3 iterations there's no more improvement, we consider that as acceptable. So this particular approach here is to optimize basically performance as a portrayal across these different modalities, in lack of a better word, classifiers in computer lingo. We were able to identify that depending on which model you choose, you get different performance metrics. They're all not 100% perfect, and they don't have to be, but they were optimized. So for example, in the initial set, we used the logistic regression model. And we had several different data sets. One was this, and we had a completely separate, slightly smaller, but still large-sized validation set to also see whether this just basically at the very top level makes sense. So when we portray this on an ROC curve, we get into the next business of how to implement it already. So the performance looks great, but where do you set a cutoff? So for example, you could set the cutoff at a score, from 0 to 1, at a score of larger than 0.9, which would be a confirmatory test. Thereby, you have probably slightly lower sensitivity, but you optimize for specificity. In contrast, when you walk the ROC curve upwards here, and you decrease the number of false positive rate here, you get a higher sensitivity, but you get many, many more false positives. Now we decided, since we want to portray it basically before the reporting decision, it's not confirmatory, it's for screening variants to improve, for example, sign out. We implemented this as a screening where we accepted that there would be some false positives, but we had at least identify a lot of variants. So once we said, all right, let's roll this out, we looked at the variants and where they fall. For example, a naive cutoff of 0.5, and then we compared cutoff of 0.25 and 0.75. Now, once we did that, we saw, well, there's obviously false negatives and false positives, and we could decide who they are. Or who, someone said, well, who was that? And that led to something which I would also consider trapped domain knowledge, which was currently, or at the time, there were 6 pathologists signing out these cases. So we just said, wow, let's feed that into the model. Now, suddenly we were not building a decision support tool anymore, but we were building a pathologist-derived machine learning model for variant reporting on a node to yes scale to compare how people were making decisions. And that led to something completely separate, which basically expanded, from the original design a little bit. And we basically said, okay, we can build an aggregate model that is great. And we can also build individual pathologist-derived models. And, for example, this is the number of variants that were reviewed. And you can see there's some sort of correlation here. But the more interesting thing is we spoke to our computer or our bioinformatics team and said, can we implement this? And they said, sure. So when we are confronted with a variant, we can basically get 6 pathologist-derived models and basically get, by hovering over it, which are the top predictors in that pipeline. So this is almost like a virtual consensus call conference, if you wish. So the interesting thing here is obviously we had to first blind the pathologist names. In the first iteration, it was very funny to give them certain names, Ben called this no, what's going on with Ben? That immediately made us realize this is obviously not Ben making the call. This is the collective experience from Ben pushed through a machine learning logistic regression classifier suggesting that Ben would likely call this, which is, creating this artificial scenario where you tend to disagree with yourself or with the model of yourself, which is quite fascinating. So, I mean, I would consider this a tangent, and I think you know what I mean with this. This was not related to what we wanted to do, but it was a very, very interesting experience. And having the ability to see what different people would choose and the differences of why they were choosing something else. Like, why was the model from pathologist 6 disagreeing with those from pathologist 2? Led us to this very interesting thing, like to drill down into the decisions of the model. So I mentioned earlier that we first decided to go for logistic regression, mostly because it assigns every single variant a distinct weight in relation to the overall decision. That's human understandable. Now, when we saw this, we said, well, wait, could we drill down a little bit deeper? So what if we not only report to the pathologist the actual decision of the model from 0 to 1, so 98 would mean 98% of the group would report this variant, and 5% would mean only 5% would report it, but then let's say, can we explore what did the machine learning model pick from the variant database to render that decision? And for that, we called that an explore function. So currently the machine learning score is portrayed. So for example, this particular variant has a score of 0.53, so 53. You could also say that it's one of the variants where the machine learning model was struggling with it, right? I think the top one here was a BRAF, and you see 92.5. But now this particular variant, when you click on it, it opens this random forest tree, and every green line represents a yes, every red line represents a no, and then each individual dot is one of the features as an output of the pipeline that contributes either by being negative or by being positive to the decision itself. So when reviewing this, you can basically drill down into the individual features. For example, that the primary caller low freak had a score of 1, and that's larger than 0.5, which the machine learning model set as the internal cutoff. So this particular implementation, which is making use of this explore function, allows a pathologist to explore why a model was actually choosing what it does. So obviously this is helpful. I think it saved several of us in certain settings of not reporting it. Clearly, you can also see that the model is struggling. So I don't say that this is like a perfect solution, but I just want to outline that this is how we implemented this explore function. Now, while this was happening, and we're talking, this was roughly in 2017, 18, we had to switch from our panel from a smaller panel to a much larger panel. This one, for example, didn't include BRCA1 and BRCA2 and ATM, but the larger panel did. So while we were validating the model from a wet lab and bioinformatics perspective, we thought, now this is a very, very interesting setting. What if we take the model from trained on version 1, and we called that, the resident model. What if we take the resident model and apply it to a completely different thing? Meaning the model has never seen BRCA1 and 2 variants, has never seen ATM variants. But what if you take that model and apply it just to this specific data set? And what we saw is the following. So the original model had a very good performance. And then if we just exposed it to the different panel and didn't retrain it, so we basically applied it as is, you could see that the performance went down by roughly 20%. Just as a matter of full disclosure, our computational pathologist team was almost protesting to do this, because this is not easy. I mean, we can talk about the details, but applying a model on a never seen before data set, they were saying, well, of course it's going to perform bad, but demonstrating it, and demonstrating that if you retrain it, that it improves, was very meaningful for two reasons. Number one is, it is clear that no model can represent the entire truth of the entire variance, available to us. So there will be, no matter what you do, you will at some point be confronted with a variant that you haven't seen before. And no matter how good the performance is, it will, if not trained with that performance in mind, likely have decreased performance. The second thing is, it said, well, if we sign out cases, we can actually retrain the model based on what we just learned. Meaning, as soon as we had more cases, so if you look at T2 retrained, we had an initial set of 568 variants. That's not a lot. But when you just even that small set of variants increased the performance quite a bit. Now, I showed you ultimately what that, if you take that second validation set on larger panel and expand this, you get to higher performance again. But the element here with this retraining or transferability experiment was to really demonstrate what happens if you're, in a way, pressure testing this model. Now, we have recently also done slightly different pressure testing. Let me quickly ask you, how about with the time? Do you have a little more? Hello, Nathan?
You have a little more time.
Perfect, wonderful. So I want to show you one thing where we pressure tested this. So as you all know, ALK rearrangements are actionable and specific therapies are available. In this case, in the meantime, several. But once this therapy is administered, at some point, resistance mutations occur. So what we thought is, if these occur in a very rare set, only 3 to 7% of patients have this, and then only a subset gets on the drug. So we wanted to pressure test whether, we had 76 tests from 50 patients with a total of these variants, and there were only 31 ALK point mutations. Now, to all the machine learning computer scientists on the call, no one would consider this data set representative for anything meaningful. This is too few data points. Nonetheless, what we thought is these are relatively specific, and these variants have never occurred before because these are effectively chemically induced. These are, evolving out of a certain evolutionary pressure under a certain therapy. So we built basically this same workflow to drill down into this machine learning element to see whether a decision support tool and a drill down function would enable us to see, can we actually identify variants that the model has not seen and that we have never seen? So what we found is, this is the list of variants and how often we have seen these in this particular data set. And there was something really, really interesting. So in the early phase, these variants have received lower scores. But as soon as we started reporting them, even the same variants, so for example, one of these, the I1171N, for example, received higher scores. So it's very interesting to see that learning is obviously happening here, And this is exactly the premise that I mentioned earlier, that under a certain experience with a certain task, the probability, and in this case, the reporting probability increases over time. And, to show you, we completed this basically on how to set a call threshold here. So we have our reported variance and the not reported variance, and we have ALK variance and non-ALK variance. So we did sort of like examine it in these 4 bins. So if you look at the ALK variance that have a score that is high or a score that's low, we had to set the threshold somewhere, resulting in, in this case, you can calculate the sensitivity. But more important here is, if you want to call this variant, obviously you can, because it's not as if the machine will tell you you have to report it or not. But I just wanted to show you this is how we thought about this very specific use case with very, very few variants as controlled by, thousands of other variants derived from our pipeline at the same time. So as an overview, some limitations and an outlook. So we have only implemented this for point mutations. And for those who have worked with pipelines, you probably know why. Index calling is very, very tricky, and getting that into a homogeneous format for your database is even problematic, whether you line at 5' or 3', how to do that bioinformatically. But it's definitely important to expand on this. We don't have other types of data in here. So we don't take, for example, histopathology or imaging data in yet, but obviously that is a big dream. We don't do tumor and normal matched genotyping. I would love to do it, but question is, what Colin, what you had mentioned is, doing both at the same time is not only good for making certain that you do proper somatic calling, but there's actually other medical or clinical utility in that. Obviously, this is powered by the prevalence in our setting. I've mentioned that we don't take a lot of breast cancers in our setting. That's more like, clinic related, but clearly, more data, etc., is always helpful. The model is currently agnostic to the medical literature. Again, we don't take into account, for example, what type of TKI or the clinical decision. So this is purely a bioinformatics and reporting tool. This is not driven by NCCN guidelines or other interfaces. We don't have this for germline, and we really want to analyze how model scores evolve over time. So if I have a few more minutes, I have a few more slides, but I think this was the blue calming slide as the end of the main part for the variant reporting.
Well,
are there questions so far?
No questions so far, but we do have about 15 minutes left if you do want to share some of your additional slides.
Of course.
Go for it.
So we have, given that by doing this, obviously we built some machine learning experience. And a few years ago, there was the sprint challenge. And some of the data that I want to show from a data set that was publicly available, which I think is a common question, which is, you find a study and you could say, oh, this is great. But the question is, like, what does that mean for my current patient? Or you could even ask, what does that mean for myself? And the principle behind, the personalized data, that the premise is that, you have a certain subset, and then by genetic or other clinical features, you want to make a decision that is most or closest matched to your patient or his or her presentation. So I think that's the big, promise of personalized medicine. Now, to me, this boils down to two things. Obviously, there has to be a certain subset that is amenable to this and to benefit from it. But ultimately, it's a data challenge. So to illustrate this slightly differently, to get back to this 80% of this problem, as illustrated earlier with the Forbes article, is actually getting the data in the right state. So what I think is currently happening, and especially for us to integrate this into a diagnostics platform, is we have these disparate data of electronic medical record, the genetic data, microscopic or other imaging data, and they're not really all on one hub. I think, our platform, I'm not sure whether we could easily transfer this to another laboratory. Obviously, our pipeline is, as many others, is home built. And even when talking to vendors and they say, oh, our pipeline works in 12 different shops, clearly that pipeline is not interoperable. It's just that pipeline from that company. The fact that it is in 12 shops by no means mean that it's completely interoperable, and interoperability with one vendor is not really interoperability. So what that is effectively, at least from my perspective, the evidence that we are in urgent need of an integrated data model. Now, every single person who invested time in building their own data model would not want to change it because it works for them, which creates this conundrum. Like, how do we want to share this, or, you know, make this more, you know, interoperable without investing some time into a data model? Now, one data model that's existing is a scientific publication, or, you know, the syntax from the HTTPS, or, you know, certain standards in a way. But how to align this across different disciplines is much harder. So one element that, you know, one of the faculty members in our group has done, and that's an amazing piece of work, was to check and test out the DICOM standard. So the DICOM standard is basically, you know, the image standard in medicine, but how to build this data model and then truly implement it, for example, with pixel data. But as such, there's no reason not to put in genomic data, which actually is, if you look at how most sequences function, is actually at some point image data that's just, you know, digitized. But this, let's just call it a vocabulary that's needed to really make things interoperable, is, I think, is a huge need. So aside from, let's say, individual efforts, what I believe is currently really needed is that we tackle this from a bigger perspective. I'm sure, you know, you heard about some of the recent FDA approvals, and, you know, obviously next generation sequencing and the associated pipelines, as well as some of the commercial vendors pursue aggressively FDA approval, and it sets a certain, I would argue, high standard that most academic medical centers may not be intuitively accustomed to. So to say that, in a way, we're also facing a regulatory sciences puzzle. And I want to show you, you know, why this is. So in some ways, innovation has to account for, let's say, the patient perspective, because you want to provide the best service possible. But at the same time, the FDA is tasked with the difficult, you know, ways and means on how to regulate this if we haven't seen it before. So what's really needed is to provide input in this as well. So at least for digital pathology and artificial intelligence, we started this work group called the Digital Pathology Alliance. It's not only in digital pathology, it's also AI and machine learning, but it primarily focuses on digital pathology because that is, I would say, the last missing image element that is just not in the medical record. And this is a group that is trying to implement certain elements that are practical and deliverable, while at the same time aiming to make it interoperable and drive this, let's just say, regulatory guidance. Now, some may say, well, I don't want to work, or I don't want to have anything to do with the FDA, which I think that's a fair position to have, and you don't have to, at least not under current standards. On the other hand, I would argue that most people who run the lab, especially the medical directors who ultimately take responsibility for the quality and safety of the patients that they test, have an inherent interest in making this safe and efficient. So whether that can be applied at a higher scale, I'm not 100% certain, but at least, you know, this is an attempt. So if you're interested, Digital Pathology Alliance, just an interest group. If you want to just join, and it's for free. And we had several meetings, and, you know, the next one is likely coming up, will be online. So please check out the website if you're interested. As a last slide that I want to show, I wanted to mention a relatively, give me 1 second. I wanted to mention, of course, the team, and I wanted to thank my mentors, as well as everyone who has helped with putting this together, as well as some of the grant and funding that I have. And with that, I'm very happy to take some questions. Thank you, guys.
Thank you so much. Wonderful talk. And recapitulating that the pathologist will continue to be needed to weed out, but we can make our lives a little bit easier by not looking at a lot of false positives. Any questions? Well, I'll just ask one. I like how you didn't just pick one machine learning algorithm and instead did this grid search over all methods, identifying sort of the best overall model. I get you said based on this F, but in reality, you were designing a screening tool where you were favoring sensitivity over specificity. Have you tried rerunning that grid search to minimize the number of false positives? I mean, you got it down to 5 in your example, but I wonder if you had a less specific model that still led to 0 false negatives.
Yeah, so we tried several things. So obviously one of the big concern here is overfitting. Like, I think it's, if you let it run more, you can probably fit it, to even, perfect, right? Like, and then overfit it to just that data set. But that wasn't the aim. We haven't done it, let's say, in a perfect way and then reapplied it or retrained it. So currently this was, what I showed you was let's say the test that we ran. The part that I would really like to do is, and I'm sure, I don't know how you guys do it, at your respective institutions, but we pre-filter variants and portray that as a variant call file, and then apply certain things. For example, base quality scores, and two callers have to call something, and that leads to a list of variants per case. Now, I know that we tried to look into it, but it's a lot of work to really take the raw VCF file unfiltered and say, can we uncover certain variants that are hidden in that, in that layer that we would never get to, that we would just consider right off the bat, too low allele frequency, too low, base quality score, or only one caller, like who would look at that? And then really try to figure out whether, despite portraying a lot of false positives, meaning with high scores, to see whether we can screen through that. We've started doing that, but it's just a lot of work. The advantage we had is that, we tracked our calls, which is pretty easy, right? Because what's reported is there and the rest is not. But really having someone to look through all the variants that were not called, we haven't done. I know that's not the correct answer to your question, but we try to do it more from the back end, not necessarily running tons of models. I think it's also a lot of work to, once you apply a certain threshold, you see performance measures, you kind of get into this realm of having suddenly 20 different performance measures to look at. And, at some point you just have to highlight one row and say, we're going to pick the one that's most sensitive. I mean, it's just pragmatic, but definitely a great point. But we haven't done that extensively or systematically.
And this is Andy. I mean, Joe, that's a great comment. I mean, we see this all the time, and even in the ovarian cancer project and our routine stuff. I mean, the challenge that we all face, and this is why so many of us spend so much time wading through pileups and everything else, is sometimes there truly is gold buried in those piles. And that's, I mean, trying to find variants that either, for example, in the data I was showing, some of those indels were very important to the biology and the predictive power of that data, yet weren't called by multiple variant callers, or they are things that are subclonal within the disease. And I mean, that is just the biggest challenge to crack. And from someone who is not a computational scientist like me, try and figure out how to make it easier on us when we're wading through that data.
Yeah, it's super, I mean, or it can be super tricky, right? Like, I don't have a perfect answer for that. But I think there was a chat question here. How do you since it's a black box, or in a way it is a black box, the question was, I think, how do you trust the model? I don't know if I'm fully portraying that right. So maybe I can just show you. There's no PHI, so I think I can hopefully share this on the screen. Let me just quickly see if this works. So, can you see my screen here?
Yes.
So this is our variant pipeline. We have a TMB counter and an MSI probability score, which is here 0. These are things I didn't cover, but if you look through this, there's a KRAS, there's a TP53, and there's a CDKN2A. Does anyone want to guess what tumor this is?
Yeah.
It's like a quiz. Yeah, it's probably pancreas, but in any case, so how this is implemented is I hope this works now, but you can see these are the paths. So if you don't trust this decision, I mean, here that's pretty easy, but by hovering over this, for example, it says the transcript KRAS is 0. The ERA ID1A is not there. So apparently the algorithm decides if it, I don't know if ERA ID1A is mutually exclusive with KRAS, but there seems to be, this is a frameshift variant, or sorry, this is not a frameshift variant, which argues for the point mutation. The GATK detection is 1, which is etc. So whether this increases your trust or let's say, how you rely on this model, we have a model ID, so we track all of this. So it's, this called master tree module is one element that is enabled here. Now the thing with the trust is once we had this data in one giant store is something completely different. So you can look at, for example, also like, I don't know, this TGF beta receptor variant is very well known to me. But once we implemented this, all of these little letters here mean, for example, this is CT means seen total how often, and how often called yes. So once we started tracking this, we basically now have, this was 4,167 times seen, and probably only called 5 times when we launched the pipeline. So this is clearly an artifact. So, some of these numbers here became really powerful. So for example, this KRAS variant, we basically call almost all of them. So once we did that, and this is just with a keystroke F, I can see all the variants that we had in this particular instance. And the interesting part here is, you see, this is by allele frequency, like where are these variants? And we can put this together in different ways, or if you want it more finely granular, you can see which cases these are. The most interesting thing is once you have access, and now we obviously left the single case level, but if you say, I want to see only the variants that are, present at 0.1, or I don't know, we have here 0.8. So the very, very low level variants, those that are present below, whatever percent in this case corrected for cellularity is, we can do something really interesting and then jump to other cases, meaning now we say, do I want to call this? And thereby just go to the next case. And then obviously that's retrieved from the database. But basically by sorting the data in a way that it is machine readable, we unlocked some of the potential, let's say just the power in that database. And I presume other folks have done similar things to build this out. So for example, you can screen a case also by just looking at the ML caller. And you can see when you sort these, you see then this MSH6 variant get a high score. But again, we've seen this all the time. So clearly the machine learning is not perfect, but that's at least how we implemented it. I don't know if this reassures or gives you a better idea how this can be implemented or not, but it's just maybe the black box is not as black anymore.
It's hands-on.
Yeah, just like
black box.
Very much.
That's a fantastic talk, and I know you were at the University of Washington not too long ago, so I enjoyed hearing some new content. And I have a similar question, just briefly. So, digital error correction models have, I'm sure you're very familiar with that, which obviously goes very well with your expertise, which is amazing. Also with some of the sort of internal frequencies. Have you implemented some of the, and there's some formal tools out there. I think we talked about this. We've tried to implement that for our cell-free DNA testing to try to get down to 1 or 2 molecules even that you're counting. Is that? Do you have any thoughts on digital error correction?
No, like ECC tools are obviously super important for, for example, bit error rates in digital imaging. I know that a lot of our imaging folks have spoken about this, and we had several journal clubs on it. And the issue here is I don't quite think that's ready for prime time clinical, right? So that's why I gave that prelude. So in the research side, definitely. I mean, people are working on that like crazy, but we haven't seen a very good use case on take that and show definitively that it's that error correction that makes it better for clinical use. I'm sure it does, but we haven't found a good, let's say, entryway to say, for example, for noise cancellation in images or so. I think I've seen several talks on that.
Well, to be clear, I'm talking about for NGS data.
Got it.
It's the same sorts of tools, but specifically, modeling the error of each nucleotide change at each position that is totally instrument and assay specific, right? It's basically what you're showing already that you're doing with capturing the internal frequencies, but just at a more granular level. So you're actually modeling a probability score, for each actual individual nucleotide change based on the sort of error probability at that. It's assay specific.
I mean, I think we haven't done any of that. We're not that far yet. The only thing that I think I've seen was, I think this was from the Google team. They have done pile-up images. I think pile-up image capture, and then applied a convoluted neural network over that. And I think that showed on the top the complete nucleotide breakdown from an IGV screen capture to say at a certain variant there are the following counts for the 4 nucleotides. But we haven't applied that. I also have to say that once we put something in clinical practice, we rarely ever change it, which is, to some people it's like, oh, this is like old and you should update it. But to us it's more like, well, this let's just call it like performance characteristics, and having those at a reliable rate builds exactly that confidence. But I asked the team, but I haven't heard on error correction yet.
Well, thank you so much for sharing your expertise.
Thanks a lot for having me.
All right.